So it's a great pleasure for me to introduce Anna Murray from Exeter, someone who I think has really led the way in the genetics of reproductive health in this kind of integrated way, thinking about the science and thinking about the clinical implications. So really pleased to hear your talk. Thank you.
Thanks, Deborah.
I'm going to have to take this with me. I've
Yeah,
had to leave
go and
it
learn
here.
how to use that. Good afternoon, everyone, and thanks so much to the organisers for inviting me. It's a real pleasure to be able to come and talk to you about some of the work that we're doing in the sort of genetics of female reproductive health. Just to start with a disclosure, I'm a co-founder of a new startup company that's working in this space, so Vartix Limited, looking at therapeutics for female reproductive health. I'd also just like to include some acknowledgements at the beginning as well, because the work I'm going to present is, as obviously with a talk of a long talk like this, it's a work of many people, particularly in Exeter. Kate Ruth, who I've worked with for I think maybe something like 15 years now. I don't like to count, do we, Kate? But Kate's here, and several others from our group in Exeter, so please catch them at the breaks. We work very closely with John Perry's team here in Cambridge, and also a lot of the functional work that I'm going to present has been led by Eva Hoffman in Copenhagen and Ignacy Roig in Barcelona. And we also have been very heavily involved with the Reprogen Genetic Consortium, the international consortium, and I've just put one of the members there, Joram Morabito, is one of the key members in that consortium who we work closely with. And I'll shout out some of the individuals as we go through for the particular pieces of work that they've been more involved with, really. So we're thinking about the female reproductive lifespan and the effect really of the ovary on determining that lifespan. So women are born with all of the eggs that they're going to have. They actually generate about 7 million oocytes at 6 months gestation. And that's the laying down of the initial ovarian reserve. And then there's stages of atresia by apoptosis, with most of those oocytes being lost actually prior to birth. So you're born with only about 1 million to 2 million eggs, and then further decline throughout life through recruitment of follicles at puberty. In fact, the ovulation process only releases about 400 or 500 eggs through lifetime. So it's this process of atresia that diminishes the ovarian reserve from that initial 7 million pool. And when the ovarian reserve gets to a level of about 1,000 oocytes, that's the point at which women will go through menopause. Saying that, we know that there's a great deal of variation at the age and the timing at which people go through menopause. And this just shows you the distribution of natural menopause in the women from the UK Biobank study. And you can see, although the average age is around 50, there's a lot of spread here. So with many women going through menopause in their 20s, 30s, 40s, right up to their mid-60s. So there's a lot of variation. And where a woman is on that distribution can influence lots of health outcomes. And this is one of the reasons that we're interested in studying this process, and particularly the timing of menopause. So we know that women with later menopause who are exposed to estrogen for a longer period of their lifespan are at increased risk of particularly some of these cancers, breast, ovarian and endometrial cancer. Whereas women with earlier menopause are at risk of other disorders such as osteoporosis, again likely to be related to estrogen exposure, because estrogen is actually beneficial for bones, but also things like cardiovascular disease, depression. Lots of things have been associated with earlier menopause. So we want to be able to understand these relationships better and really give better risks for women of their chances of developing these particular health outcomes relative to their reproductive lifespan. The other key area, of course, the key sort of health outcome related to ovarian reserve and reproductive lifespan is fertility. And there are very close links between the timing of menopause and natural fertility. You can see in the plot here on the right-hand side that decline of oocytes throughout life and how that maps onto fertility levels. And we know that women, if they're going through menopause around the age of 50, will become naturally infertile at around the age of 40, so about 10 years prior to menopause. And of course, this is really important, particularly in modern populations when women are delaying having children till later in life. And so this can have a very large effect on the chances of conceiving naturally, and goes alongside the increase that we've seen in use of in vitro fertilization technologies. When we look at that distribution of age at menopause, we know that about 10% of women will go through menopause before the age of 40. And this is often defined as a clinical condition of primary ovarian insufficiency, or POI. And this is something I will come back to a few times during the talk. And in that population from the UK Biobank, we have about just over 2,000 women who meet that criteria to have menopause earlier than the age of 40. And these often seek clinical help for hormone supplementation and perhaps trying to conceive through IVF and so on. So this is actually a large proportion of women that fall into this group. And the etiology of that clinical condition is an area of a debate, and certainly has been an area that's been studied extensively by genetics. And I'll talk about that as we go through the talk. It's not all about genetics. Quite a lot of what determines the timing of menopause are the non-genetic factors. So here is just a list of a few things that have been associated with menopause timing, the non-genetic factors. I think probably the best predictor of when somebody will go through menopause currently is their mum's age at menopause. And obviously this is probably partly determined by their shared genetics, but also by their shared environment. Probably the most robust non-genetic associated factor is smoking. We know that women who smoke go through menopause on average 1 to 2 years earlier than non-smokers. And there are various other things that have been linked with various levels of evidence. So we think about 50% of the variation in menopause timing is about genetics, and about 50% these non-genetic factors. So I'm going to talk about those genetic aspects. That's the area that we've been studying extensively in my group in Exeter. And I'm going to divide it into the types of genetic variant that we've been interested in, because this is often how we think about it and think about the genetic architecture of a particular trait. So menopause timing is what we call a classic multifactorial trait. So it has a mixture of genetics and non-genetics. And in many cases, it's multiple genetic causes, not just a single one. And so we're going to start by thinking about the common variation in the genome. These were probably some of the easier things to assess at scale in large population studies. So these are variants that are present in about greater than 0.1% of the population. And they tend to have very small effect sizes, though. And we find these through genome-wide association studies. And this is the largest genome-wide association study published to date that's come from the Reprogen Consortium, but led by our group in Exeter and a group in John Perry's group in Cambridge, and the sort of lead analysts here, Kate and Felix, both here at the meeting today. And we found that there were 290 independent genetic signals associated with age at menopause. And this was published in 2021 now in Nature, which is really exciting, and we had the front cover of Nature as well. So that picture there is our front cover talking about our study. So it's really exciting for women's health conditions to be on the front cover of Nature and getting these high-profile publications. And what we found from this big GWAS was that the effect of each individual variant varied from really small effect sizes of just a few weeks up to over a year. 17 months was our largest effect per allele on the timing of menopause. We were able to replicate these findings in an independent study. We looked at the 23andMe direct-to-consumer company data, and we replicated these initial findings. So we think these are really robust associations, and they also replicated, the study was in European individuals, but we replicated broadly in East Asian women as well. Saying that, we found these 290 signals really exciting, but we only explain about 10 to 12% of the variation in menopause timing with those signals, although that's about 32% when you take the whole of the common variation in aggregate. So we're getting there with trying to explain the heritable component with these common variants. But what you can do with these variants is really start to understand more about the biology of these processes. And this is where one of the real values we found from this initial study. I'm hoping you can see this fairly clearly. So what we found was, in those 290 signals, about 2/3 of them were in or near to genes that were involved in DNA damage response. Now, the role of DNA damage had been highlighted before in ovarian aging, but I think what this study really highlighted to us was the extent of it. And it wasn't just one or two processes, it was multiple processes that were all involved in repairing, surveilling, dealing with damaged DNA in the body. So on this slide, you can see in the top here the various DNA damage repair processes, and these are the genes that we highlighted and where the type of processes that were affected, that they're involved in, and that we think are being involved in ovarian aging. Two really key proteins, or two key pathways, are these two shown at the bottom here. So this one here, the CHEK2 ATM pathway, and this one, the CHEK1 ATR pathway. And these are involved in repairing double-strand, in this case, and single-stranded breaks. And in these pathways, all of the genes here that are in this blue colour were genes that were highlighted from our genome-wide association study. So you can see it wasn't one or two. The majority of these genes in this CHEK2 pathway are coming up as being, small variations in them are influencing the timing of menopause. And similarly for this CHEK1 pathway. So this is really showing us that these pathways are really important. And about 58 of these 290 genes, so a good proportion of them, were involved in the apoptosis pathways, so clearing those atretic oocytes in the ovary. So this kind of just summarises what we found from that biology part of the study. So if we map that onto the diagram I showed you at the beginning, where we saw the establishment of the reserve and the depletion, and we were able to look at expression of these proteins and the types of cells they were expressed in. And we see that actually these processes are occurring throughout life, and we found genes that were acting throughout life. So there are genes that we think are influencing the establishment of this pool, and then all of these atretic processes, the apoptosis throughout life. So we have genes that are involved in double-strand break repair, specifically in meiosis. Now that we think the recombination process of meiosis, where you're generating breaks and then recombining the DNA, that is a key process. And then, as I mentioned, the apoptosis as well. So that's the sort of the biological insights that we can derive from these genome-wide association studies. One of the other things that we were able to do was to look at whether we could predict early menopause in women by combining these genetic variants together. This would obviously be a really useful thing to be able to do, if, particularly with the links with fertility, if we could predict which women were likely to have an earlier menopause and therefore likely to hit infertility at an earlier age. And so we can combine those genetic variants, in this case the 290 variants that we identified from the GWAS, into a genetic risk score. So here you're looking for each woman, you're giving her a score based on which version of the genetic variant they have, and then how big the effect that variant is on menopause timing. And here in the two colours, you can see in blue are women who had menopause under the age of 45, and in red, women who had menopause over the age of 45. And you can see that there is a shift in the average genetic risk for the women with earlier menopause compared to the women with later menopause. So obviously, we'd like there to be a lot more separation between the two if you want to use this in any sort of predictive clinical context, but there's certainly some predictive power here for these genetic variants in combination. Another way that we looked at this was to think about, could we predict that clinical category, that POI or menopause under the age of 40 group of women? And here we took the genetic risk score, and on the x-axis is the percentage of genetic risk score, percentile, sorry, and their odds of having POI. And when we looked at the top 1% of genetic risk carriers, we found that these women were 5 times more likely to have menopause before the age of 40. So again, this is actually becoming more useful, perhaps in a more clinical context. So we thought this was an important finding as well. One of the other things that we were interested in looking at, and we used this genetic risk score for, was to look for new mutations. Now, I mentioned that this DNA damage repair pathway is really, really important in this trait. And so we predicted that, that if there were variation in that DNA damage repair, that perhaps there would be more genetic mutations passed on. And we looked at this with a group led by Hilary Martin from here at the Sanger Centre in Cambridge and Chin Huang, and they had access to data from the 100,000 Genomes Project, the GEL project, where they have 8,000 parent-offspring trios, and you need these trios to be able to determine which variants arise de novo in the offspring. What Hilary and Chin were able to do was to look at the genetic risk score that both parents were carrying and see how likely it was that the offspring had a new mutation. And what we found was that it was only when the mother had a high polygenic score for earlier menopause that we saw a significant increase in de novo mutations in the offspring. So if the father had the same genetic score, he did not pass on more de novo mutations. And to give you an idea of the size of that effect, so for 1 year earlier genetic risk for menopause in the mother resulted in 0.074 more de novo mutations in the offspring. So it's a small effect, but we try to do a back-of-the-envelope calculation to give a better sense of that. So if you think about the genetic risk score that somebody with POI has, they're likely to transmit 1 extra de novo mutation to their offspring. And that's actually a bigger effect than the effect of smoking on de novo mutations. So this, relatively speaking, is a substantial effect on the rate of de novo mutation. So this was a really exciting finding. So moving on now to a different trait associated with menopause, and that's menopause symptoms. Now, we know that there are many, many symptoms that have been attributed to the menopausal transition, and particularly at the moment it seems to be a very popular topic in the media and so on. And there's probably 50 or so different symptoms that have been attributed to the menopause, and some of those I've listed on this slide here. Probably one of the more common ones is hot flushes or vasomotor symptoms, and probably more than about 75% of women will experience these, and they can be really debilitating. They can stop women wanting to work, to interact with friends and partners and family, and can last more than 10 years in about 10% of women who experience them. So we wanted to see if we could use genetics to understand more about the biology of these symptoms. So the first symptom we looked at was this vasomotor symptoms, and this is work very much led by Kate in Exeter, and there's a link to her publication here. And she again used the UK Biobank data, and she used the health record data to pull out cases of women who had experienced hot flushes or vasomotor symptoms. She had 14,000 cases in the UK Biobank and 77,000 controls. When you do this analysis, this association analysis, she found a single signal, which was at the TACR3 gene. So for those not familiar with these plots, these are the genes along the bottom here, and the dots represent the variants from the genetic association study, the height of them on here, the strength of the association. So this is a minus log of the p-value. So this is the strongest signal here. And all of these are correlated with it, and so that's really a single signal. Now, this is a gene that's actually well known in female reproductive biology, because this gene, when it is lost, when you have a loss of function variant in this gene in the homozygous state, can cause puberty not to occur, so hypergonadotrophic hypergonadism. And there are also signals in this region associated with the timing of menarche, so puberty timing, and that's shown by these triangles here. So this shows you the variants that are associated with menarche, and these are the loss of function variants in this gene. And as you can see, they are completely flat. They're not associated with having vasomotor symptoms. So it seems that this signal that we're seeing for vasomotor symptoms, although it's in this region that we know is important in these similar conditions, if you like, it's actually separate. It's a different signal. I said this was a well-known region, and in fact the TACR3 gene codes for the neurokinin 3 receptor, which is in the arcuate nucleus of the hypothalamus, and it stimulates the neurokinin B gene. So it's expressed here by these candy neurons, and it's involved in initiating puberty and signalling to the ovary through gonadotrophin-releasing hormone during menstruation. But we also knew before we did our genetic studies that the lowering of estrogen at menopause actually causes hypertrophy of these candy neurons, and that this increases signalling or expression of this NK3B and stimulates this thermoregulatory center here. And it's thought that this is what triggers the vasodilation and vasoconstriction that women experience in hot flushes. And because this has been known for some time, it's the target of some clinical trials. And in fact, Julia Prague, who's an endocrinologist who works with us in Exeter, led one of the early clinical trials here published in The Lancet targeting this neurokinin 3 receptor with an antagonist, and showed improvement in patients for their hot flushes. And there's a drug been released, I think in the UK about this time last year, fezolinetant, which is also an NK3R antagonist, which is for the treatment of hot flushes. So this really fits nicely with the genetic data that we're seeing with the TACR3 region being really important. So we wanted to pursue this and work more on this area, find out more about the mechanism, and also hopefully to find some new signals. So that's a project that's ongoing in our group, and the person leading the analysis on this is Alok Singh, and Alok's in the audience today, so please chat to him about it in the break if you find him. And what we're doing is, this is a project funded by the MRC. It's a project that Martha's involved in as well, and several others. And what we're trying to do is to increase our sample size for vasomotor symptoms, and this shows you the results from the unpublished results from the ongoing meta-analysis of vasomotor symptoms, and we have over 160,000 cases now and 300,000 controls. Slightly disappointingly, there's only one really strong signal, and surprise, surprise, that's the TACR3 signal that we already know about. But we think there are a few things creeping over the genome-wide threshold line, so we're optimistic that we'll be able to find some novel signals. But it's certainly clear that that TACR3 signal is the predominant one. And then what else we want to do is to try and identify associations with the other symptoms associated with menopause. So the plan is to look at some of the things like poor sleep, some of the sexual dysfunction, and potentially some of the things called brain fog. It'd be lovely to try and understand what that is, if that's a thing. And we can use genetics to look for causative mechanisms and see if those mechanisms overlap. Are there different etiologies? Are they all part of the same condition? So that's the plan ongoing. And we're just at the start of this project. We have another 2.5 years to go. So moving on to the next category of genetic variant, which are the low-frequency variants, or the rare variants. And these occur at less than 0.1% of individuals in a population. And these are ones where they have a more moderate effect size. So to be able to find them at that frequency, they have to, by definition, have a bigger effect size. But you can't find these rare variants using those SNP arrays that we've used for GWAS. The technology is just not good enough to be able to robustly say that something has genotyped correctly. So we have to use a sequencing approach. And of course, with sequencing, it then becomes more expensive. So then to be able to do this at scale, to find these variants for the general population, this has had to come about with the larger population studies really providing both exome and genome sequencing to be able to detect these rarer variations. So we started off by looking in the UK Biobank exome sequencing release a few years ago now, And so the exome only accounts for about 1 to 2% of the genome, but these are the protein coding regions of the genome. And we looked at two main types of variant. We looked at variants that when they were present, they cause loss of the function of the protein, so complete loss of the protein if there was a gain of a stop codon, for instance, or a splicing variant. But also we looked at changes in the amino acids, so the missense variants as well, and we categorised them depending on their predicted pathogenicity or their deleteriousness. So we tried to pick the ones that were more deleterious. And as well as looking at individual variants, to increase the power of the method that we were using, we grouped variants together, and we grouped them according to their predicted effect. So we grouped the loss of function variants together into a burden test, if you like, and also the deleterious missense ones. And then we also did a test where we put all of them in together. So we did 3 different types of test, as well as the single variants. This work was published in Nature just at the end of last year, and the lead author, Stasha Stankovic, is in the audience as well today, was doing a PhD at the time. And I'm just going to give you a flavour of some of the results from that study. So the exome sequencing identified 9 genes where there were rare variants associated with timing of natural menopause now. And you can see, to orientate you in the forest plot here, the blue bars are the deleterious missense variants, the red the loss of function, and the combined together all damaging in black. If it's a solid dot, it reaches our p-value threshold, below our threshold, and if it's an open dot, it's not quite significant. So you can see from this that there were some genes that we had recognised from our GWAS studies, CHEK2, for instance, which increases menopause timing for the loss of function particularly. BRCA2 we'd identified in the GWAS as well, variants near BRCA2, which is reducing timing of menopause. But there were these 5 genes that we hadn't identified before, and they're highlighted here. Some of those, ETA1 and PALB2, we put into the DNA damage response pathway of genes. And of course, the beauty of doing these studies now, unlike GWAS, where you're just identifying a region of the genome, here you're actually much more confident that these are the genes that are actually causing the effect on menopause timing, because you're actually looking at damaging variants within those genes. These 3 genes are not in the DNA damage response pathway particularly, and I'm going to focus on these 2 to give you a little bit more information about them. So the first one, SAMHD1, was really interesting. So again, like CHEK2, its loss of function variants and damaging missense are increasing menopause timing. And this was a gene that's well known to the cancer biology field. It's a gene that's frequently deleted somatically in cancers or mutated. And obviously, when you look at this gene list and our GWAS list, a number of these genes are actually well-known cancer genes. So we looked at all of the genes and their association with cancer in the UK Biobank. And here are the results, and for 4 of those 9, we saw significant associations with cancer. And here we're looking at males and females and both sexes combined. And the cancers are any cancer, but also blood cancer and the hormone-sensitive cancer, so things like breast, ovarian, endometrial, and prostate in the men. And you can see that for these 4 genes, these are, I think, well-known associations with cancer risk, but SAMHD1 at the time that we were doing this study hadn't really been identified as a predisposition gene for cancer. As I said, it was mutated somatically, but not as a predisposition gene. So this, and there's been a couple of other studies now that have shown similar findings. So it looks as though germline variants in this gene are associated with increased predisposition to cancer. So again, there are very strong links between the genetics are showing really strong links between the biology of cancer and the biology of ovarian decline and reproductive aging. And that was summarised really nicely in this News and Views article that went alongside our paper that came out last year. And I just wanted to highlight this because I really love the diagrams that they've used in this paper by Anna Gorley, and she has illustrated that really nice. So these are the 9 genes ordered actually by direction and effect size. So you can see we have genes where loss of function is reducing menopause up to increasing menopause timing, so from around about 6 years up to about 2.5 years. Yet when you look at the cancer predisposition genes, all of them are increasing the risk of cancer. So they're having opposite effects on menopause timing, but they're all having the same direction of effect on cancer. And when you look at what those genes are doing, there seem to be two broad pathways. So there are genes like BRCA2 and PALB2, where these variants are involved in the repairing damaged DNA. So you would imagine that by loss of these genes, you're having more damaged oocytes, and so these are getting depleted faster, and therefore you result in earlier menopause. Whereas variants in SAMHD1, CHEK2, HELB are detecting damaged oocytes. And so if these aren't present or working correctly, those damaged oocytes are able to survive longer, and so you have a slower depletion of ovarian reserve, resulting in later menopause. So it was a really nice way to depict it. So we've actually been able to look at these sorts of things in mouse models, and by we I mean Eva and Ignazi have done these mouse studies, and these results were actually in the GWAS paper. And what they did was to look at two of the key genes in those big pathways that I showed you at the beginning. Remember, we had CHEK2 and CHEK1. CHEK1 actually wasn't one of the GWAS hits, but it was the central player in that whole pathway of genes, that ATR CHEK1 pathway. So when you look at the CHEK2 knockout mouse compared to the wild type, in the young mice, there is actually not much difference, no statistical difference in the number of follicles present. But when you age those mice up to 13.5 months, and I'm told this is a very old age for a mouse, you start to see that difference, with the CHEK2 having more total follicles than the wild type. And this fits really nicely with what I've just been describing. So we think that loss of CHEK2 reduces the amount of apoptosis or clearing of those oocytes. So you actually, in adult mice, have more oocytes, and that means they can extend their fertility. And we know that they extended their fertile lifespan by about 20%. We weren't able to do similar experiments with CHEK1 because the CHEK1 mice are not viable, knockout mice are not viable. But we were able to look at overexpression of CHEK1 by having an extra copy of CHEK1. These are called super CHEK mice. And when you look in the young mice in this case, in contrast to what we saw with CHEK2, we saw that the super CHEK mice did have more oocytes in the early stages, and they maintained that difference in older mice, but it seemed to happen much earlier than for CHEK2. And again, that fits with CHEK1 affecting that repair process early in life. So you actually, in the young mice, have a larger ovarian reserve, which persists into adult mice. And again, they produced normal live pups and extended their reproductive lifespan. So you can see that we have two different mechanisms that are both leading to the same outcome, and that it's really exciting that we're starting to be able to sort of manipulate these genes and influence reproductive aging through these different mechanisms. And by understanding the mechanisms, we can really get a handle on how we might manipulate them. The other gene that came up that I'd really like to mention is this one, ZNF518A. And this was a new gene to us. We didn't really know much about it. And in fact, there's not much about it in the literature either. It's a gene that's a transcription factor. It binds to about 18,000 sites in the genome, and work that sort of some in silico work that Nick Owens in Exeter did a lot on what was known sort of from in silico data. He was able to show that these binding sites, these 18,000 binding sites that this protein binds to, are actually enriched for the GWAS hits that we had, those 290 GWAS hits. So that was really interesting, that perhaps it could be regulating multiple targets that are important in the ovary. This shows you the forest plot here of the association with menopause timing. So it actually has the largest effect on menopause timing that we've seen to date. So loss of function carriers, although they're rare, they have menopause about 6 years earlier than non-carriers. And really interestingly, when we looked at the puberty timing data, so menarche timing, those loss of function carriers had later menarche. So actually, these variants are reducing the reproductive lifespan at both ends, if you like, by delaying menarche and reducing menopause. Adjuna in the Copenhagen lab looked at expression in various oocytes, various stages, and the granulosa cells in the ovary, and showed that it's expressed in these in the germ cells, and it's expressed at lots of different stages. So this is a really exciting finding, and I can't really tell you what the answer is, and this is the focus ongoing work. We have John Perry, myself, Eva Hoffman, and Steve Franks, who works on polycystic ovary syndrome at Imperial. The four of us have a Wellcome Trust Discovery Award, where we're going to really try and take genes like this and understand exactly what's going on. We have 6 years of funding to develop some model systems in both cells, organoids, and model organisms to start to understand what these genes are doing regarding ovarian aging. So that's really exciting. And the ongoing work, one of the ongoing projects in our group is to move on from the GWAS and the exome sequencing and to start looking at the rest of the genome, which is obviously a much bigger task. So in GWAS, we are covering the whole genome, but you're actually only detecting about a million of the genetic variants, and it's the common variants that you're looking at. In exome sequencing, you go much smaller, much lower frequency, of course, but it's only the protein coding region. So in genome sequencing, we can go through to all 3 billion bases, but it comes with its challenges, of course, not least of which is having the data to be able to do that at scale. But here we're helped by the fact that the UK Biobank has released in the last year or so whole genome sequence data for all 500,000 individuals. The All of Us study in the US will, we're told, hopefully still, have 1 million individuals with whole genome sequencing, but there's certainly data on 250,000 at the moment. And we've also used the TopMed study as well that has about 130,000. So this is a really fantastic resource. As I said, this makes, the challenge is how you deal with it, how you work with it. And we have a really talented team of people working on this project, on actually the nuts and bolts of getting a framework together to make this work. And it's led by Mike Whedon and Caroline Wright, but lots of other talented people who can really work with these data. And this is a really brief overview of how the pipeline works. So they've developed it in UK Biobank initially, and they can take the data on just over a billion variants, do single variant association. And then what we're doing is aggregating those, a bit like I said for the exome sequencing, where we took the functional variants, and we certainly do aggregate them as coding sequence as well, and we're picking up more things in that because we're actually covering a bit more of the exome with the genome sequencing than we did previously, so that's definitely important to do. But the key thing is that we have these regulatory burden tests as well. And we take various ways of annotating these, but we can look at predicted enhancers, promoters that are proximal to genes, are within genes. And we also do it sort of agnostic to function as well, just taking sliding windows of 2 kilobase pairs across the genome. So here we're sort of lumping these rarer variants together to see if we can really identify regions of the non-coding genome that are important. And these are some, again, unpublished, very preliminary data that the Marina, who's in our team, has been working on this project with respect to ovarian aging. She has a poster at the meeting, so please go and talk to her if you want to find out more about it. This is just a summary of her findings. So because of the multiple testing problem that you have when you're doing the whole genome, it's actually really hard to detect positive signals. But you can see these are the things that have come up as significantly associated with age at natural menopause. You can see the coding things, some of the things we know, the CHEK2, missense and loss of function. And many of the genes we've just mentioned, there's ZNF, for instance. But here we had a few more carriers, as I said, because we were able to capture the sequence better, I think, with the whole genome. But most excitingly, we have these 2 regions that are in the non-coding region. We've still got to work out what they're doing and follow it up further, but we are able to start to find these regions which are really hard to find and to try and understand them better. So to finish off the last few minutes, I want to talk briefly about the rare variants. So these really are the oldest category of variants that we looked at, really. We started to look at these many years ago, before I was even working in this field, actually, using family data and looking at co-segregation of rare single gene causes of predominantly premature ovarian insufficiency. And there have been many publications in this field. It's just a smattering of them, and many genes, more than 100 single gene causes of primary ovarian insufficiency have been reported. And that's in papers like this. So this is a pedigree showing you a dominant cause. You can see 3-generation family, lots of people carriers and affected. So this gene here, you know, a really robust association in this particular instance. But you can see you need big families, and obviously this is a condition that is affecting fertility, so it's difficult to find these. This is an example of a recessive mutation in MCM8 in a consanguineous pedigree, and here you can see 3 carriers, 3 affected women who had primary amenorrhea, so they never got to menstruate, and that's, if you like, the extreme case of the extreme end of the menopause spectrum. So there are definitely examples in the literature of really robust single gene causes. I worked on one called the FMR1 gene. This is how I first got interested in this field, actually, when I was working in Southampton. And we actually showed that a variant in this FMR1 gene called the premutation was associated with menopause timing and, in fact, increased risk of POI. And we showed that it was the premutation category, and it's a gene that has a CGG trinucleotide repeat in it, which when it expands up to more than 200 copies becomes methylated and causes the Fragile X syndrome. But we found it was associated with this very early menopause phenotype. And when we looked in those families and compared in the blue here the premutation carriers to the orange are the full mutation carriers and the non-carriers, we basically saw a shift in menopause age of about 5 years for these premutation carriers earlier. And we did a similar study going out to the population. This was a study in the Breakthrough Generations study. We had 2,000 women where we genotyped for this repeat. It's quite difficult to capture by some of these methods, until recently, in fact, with non-sequencing methods. And we saw, again, that there's about a 5-fold increased risk of POI if you're carrying a premutation from the population data. So it's kind of a halfway house between those common variants and the single gene causes, but it's definitely robust. And we wanted to look at the penetrance of those single gene causes in the population. And by having the data in the UK Biobank, we're really able to do that. And this is a publication that came out a couple of years ago now in Nature Medicine. And we looked at genes that were on the Genomics England panel app for POI, so these sort of clinically testable ones, 67 of them. We also added some from the literature that we knew about. And on this slide, I'm showing you the 37 genes that supposedly have a dominant mode of action. So a single variant, heterozygous in these genes, deleterious variants, should cause POI, according to the literature. And as you can see, so these are, this shows you the number of carriers who have loss of function variants in this gene, and this shows you their menopause age. So you can see, for real surprise to us, we were not expecting to see this, that in every case, there are women who have menopause over the age of 40. In fact, most of them do, and the average age is not much different from 50 for most of these carriers. We definitely do see people who have menopause before the age of 40, but not really more than you'd expect by chance for a very large majority of these. This is comparing to people who don't carry variants in any of these genes. Obviously, you expect 1% of women to have this. Sorry, I've got to go. I'll skip that one for now. And so we also looked at whether these genes might be a risk factor rather than a single gene cause, and we did find that 5 of them were. Again, some of them we knew about, but some of them were novel here. So again, it's more about being a risk factor. So we feel that the POI clinical condition is not as simple as being a monogenic thing. It's probably much more likely to be polygenic, oligogenic, with just a few cases that are monogenic, and perhaps more likely to be recessive, actually, than dominant causes. And I bring you back to our polygenic risk score model, where we saw that those at highest common variant genetic risk were much more likely to have POI. So to sum up, Our genetic studies are really highlighting the biology of these processes and that DNA damage repair is absolutely fundamental for reproductive aging across the lifespan. That we can use genome sequencing to really get down to the causative genes that have large effects. We can manipulate these genes and therefore extend reproductive lifespan in some of these model systems. And that these single gene causes are much more likely to be rare for causing early menopause, and that's more likely to be a polygenic condition. Just showing some nice pictures of Exeter/Devon, for anyone who's interested to visit. And I'll just finish again with my acknowledgement slide. So thanks to everyone who contributed. Thank you.